Okay, so in this video, we're going to be talking about transcription and translation. So they are the steps that help our cells produce proteins. So let's start with RNA transcription. This is the process where DNA is used as a template to create messenger RNA, or mRNA. The first step is to access the DNA inside the cell's nucleus. Step 1. DNA unwinding. So the enzyme helicase unwinds the DNA double helix, exposing the genes that need to be copied. Step 2. RNA polymerase. RNA polymerase comes into play. This enzyme binds to the promoter region of the gene. It then moves along the DNA strand, reading it and synthesizing a complementary RNA strand. As RNA polymerase moves along the DNA, it adds RNA nucleotides. Again, the four nitrogenous bases: adenine, uracil, cytosine, and guanine. Remember, it's uracil, not thymine, because this is RNA we're talking about. And so, it starts adding these nucleotides complementary to the DNA template. And by that, I mean A goes with U, C goes with G. Step 3. mRNA completion. Once the RNA polymerase reaches the end of the gene, it detaches, and the mRNA strand is released. This mRNA is the blueprint that will be used to make proteins in the next step, translation. Step 4. RNA translation. This process occurs in the cytoplasm, where the mRNA interacts with ribosomes to create proteins. Step 1. mRNA enters the ribosome. Once the mRNA leaves the nucleus, it attaches to a ribosome, the molecular machine responsible for protein synthesis, or the creation of proteins. Step 2. tRNA and codons. mRNA is read in sets of 3 nucleotides called codons. Each codon corresponds to a specific amino acid. Transfer RNA, or tRNA, has an anti-codon that pairs with the mRNA codons, ensuring the correct amino acids are brought to the ribosome. Each tRNA molecule carries an amino acid and matches its anti-codon with the mRNA codon. For example, if the mRNA has the codon AUG, then the tRNA will bring the amino acid methionine. Here is a wheel that shows what each codon codes for. So you would start in the middle for the first letter. Step 3. Protein chain formation. The ribosome links the amino acids together in the correct order, forming a polypeptide chain. This chain will fold into a functional protein. As the ribosome moves along the mRNA, amino acids continue to be added, one by one, until the entire protein is made. This process ends when the ribosome encounters a stop codon, signaling the end of translation. So that's how transcription and translation work together to produce proteins in our cells. Transcription creates the mRNA copy of the DNA code, and translation uses that code to build proteins, the essential molecules that perform countless functions in our bodies.